Christmas lectures for young people, lecture 2. "20 seconds. Ten, 9, 8, 7, 6, 5, 4, 3.
Life depends on a lot of chemical reactions which are going on in your bodies and no doubt in the chickens' bodies all the time. Whether that be digesting the food that you're eating or building up the things that you're made of, chemical reactions are going on all the time. And all of them that go on in your body are controlled by proteins. And these proteins are acting as protein catalysts. What are catalysts? Well, catalysts are substances which make chemical reactions happen faster than they otherwise would without being changed themselves. They change the rate of the chemical reactions. And proteins, and these particular proteins we call enzymes, are controlling all these chemical reactions going on in your body. So let's wonder about a catalyst. We can start by thinking about a substance which you're all quite familiar with, especially if there happen to be any of you who didn't like the color of your hair and decided you liked to change it to a fair color. If you want to bleach your hair, you know you use a substance, hydrogen peroxide, which is quite a familiar substance. The molecule of hydrogen peroxide is a simple one, which looks like this. It's just 2 oxygen atoms joined together and 2 hydrogen atoms attached to them, but it's really rather active. And you will know that although it looks like a stable liquid, rather like water, if you put hair into it, then the hair is bleached, and if it goes in brown, it comes out almost white. So it's a rather active substance biologically, and you can well imagine, I think, that if that got into the wrong place in your bodies, that would be rather dangerous, and you might like to get rid of it. And indeed, it is dangerous, and you do have an enzyme, a protein catalyst, that helps you get rid of it if it should accumulate anywhere. But there are other substances which act as catalysts to help decompose hydrogen peroxide, and we can have a look at one of those now. One of these substances is manganese dioxide. Now that's a black substance, a black powder, and Bill here has got some manganese dioxide in the bottom of this conical flask. And along with the manganese dioxide, he's got a test tube full of hydrogen peroxide. And if he shakes up the flask to mix the two, you'll see that something happens. Missed.
That's right.
Well, what was happening was that the manganese dioxide helped the hydrogen peroxide to decompose rather quickly. And that's a useful reaction. You could use it in driving a rocket, for example. Well, you don't have manganese dioxide in your body, but you do have enzymes that affect the same thing. And all creatures have this, and some of them use it in a rather spectacular way. For example, there's a particular little beetle known as the bombardier beetle. And here's a specimen from the Natural History Museum, in which you can see this little beetle. It's about 1 centimeter long, and it uses this reaction to create a jet of steam and oxygen, which squirts out poison at any enemy that feels like attacking this little beetle. So it looks small, it is small, but it's quite a powerful little beetle. Now that's a bit too small to see, but Bill has a pet beetle of this sort, which he's bringing in here, and it's quite friendly with Bill, but I have to be really rather cautious about how I approach it, and
would you like to stroke it?
I can try to stroke it, but it doesn't like it very much.
I think I better
Well,
take that.
you'd better take it away again, Bill. This beetle has means of protecting itself, as you see. Now, as I say, we don't do the reaction quite as vigorously as that, but it is going on all the time in us and in other living things. And now what I'd like to do is have a little experiment to see whether we can find this enzyme, this catalyst, which breaks down hydrogen peroxide in some material from living creatures. So, thank you very much. First of all, we'll have a look for it in liver, and there's a nice plate of liver, and we'll have a look for it in some celery, and here I shall need some help. Now, can we have some volunteers? Well, there's a girl on the corner there that looks, and the girl behind her there, that looks nice, and there's a little boy on the end there. That's fine, you. Can you two come over here with you? Well, I think, yes, you can come here as well, and you with this one with the celery. Now, we want somebody else. There was a volunteer around there. Yes, you'll do fine for a volunteer here. Right? I think that will just about do. Now, what I'd like you lot to do as quickly as you can is to cut up that liver into small pieces. So quickly, set about chopping it up into small pieces, and don't mind if you get it on your fingers. Now, would you break up the celery and put it into this blending machine. Right? You could give me a piece to chew while you're at it, because it's really quite good celery. Now you need to chop it up as quickly as you can and as fine as you can into small pieces. Chop, chop, chop. Now, as they're going on chopping, well, I think that will do there. But not you. You've got to work very hard at that liver. Then I'm going to give them each the same quantity of water. So you're going to put the water into the blender there. And when this liver's been chopped up a bit smaller, we're going to put the water in with that as well. So would you like to put the water into the blender and put the lid on? And then we'll switch on the blender and start grinding it up. And how's the liver coming on? Well, it's looking pretty messy. Yes? Would you? Yes, keep on chopping it up. Yes, it's getting to look better. You're digging the enzyme out of it if there's some there. All right? A bit more, another chop. Right? You're getting a bit grubby, are you? Yes, it's... All right. Well, no, we're going to wash that off in a minute because we need that. You mustn't take that away with you. That's important enzyme. All right, how's that coming on? We're about ready. Well, I think you'd better leave off there, but keep your hands there. Now hold your hand over the plate, and I'll pour some of the water. Hold your fingers down, and we'll see if we can wash some of that off with the water. All right, and yours. All right, now sort of stir that up with the liver a bit. All right. Yes, it is a bit like that. All right. Now, see if you can pour all of that back into the beaker and see. All right. Careful, into the beaker, not onto the... There's a bit more. Yes. Well, perhaps we need a mop to mop this up with. That's, a lot of the enzyme has fallen on the... All right, there's that. Thank you. We better take that plate away. All right. Now, can you pour that into the beaker that you started with? Now. Well, that's fine. Good. All right. Now then, if you stand both behind these two cylinders, you over on that side, and you, this team on this side. All right. Now, you hold your celery juice, and let's have someone with the liver juice. All right. Now, these two cylinders are marked celery and liver, I hope, and Bill is going to put hydrogen peroxide into them. There's some hydrogen peroxide in the first one, and here's some hydrogen peroxide in the second one. Now then, I'm going to say, don't do anything yet. I shall say 1, 2, 3, go, and when I reach go, you each pour the juice into the cylinder. All right? And we are going to see whether anything happens, and if it does, which reaction is the strongest, and there'll be a way to tell that. So are you ready? Get into position then. 1, 2, 3, go. Well, the liver started well, but it's going to lose. The celery is coming up now. It looks to me as though the celery is going to win. Yes? I think the celery will reach the top. Yes, it will. It's getting to the top. I think it might even overflow. Yes, the celery got to the top. Well, I think we should cheer the celery team. Thank you all very much. Well, I think it's fair to say that it was easier to grind up the celery than it was to cut up the liver. Perhaps that had something to do with it. Anyway, if we go about this in a very scientific manner, one can isolate this enzyme from celery and liver and so on. And when we do that, what we get is a nice crystalline powder. I can pour some of this onto this watch glass. There you are, a nice straightforward looking chemical substance. And that's an enzyme catalase. And of course the same sort of thing should happen quite well with a purified enzyme. So if I take some of this, not very much, and I dissolve it in this water. Now I'd better not stir it with the spatula, had I? And stir it up. And I put some hydrogen peroxide into the cylinder. Like that. That's again not very much. And then I add the enzyme to that. I should get a satisfactory reaction. There you are, Bill. You better. So you see, in our bodies, we have some enzymes which will at least decompose hydrogen peroxide for us. And our question today is, how do these protein molecules, these enzymes, act as catalysts? What's the basis of that? Well, that's been thought about, of course, for a long time. And the accepted view for quite a long time is that an enzyme molecule has a particular shape. And that's the important thing about it. It has a quite definite shape in 3 dimensions, like a big jigsaw puzzle piece. And the molecule that it's going to interact with has a shape like the other jigsaw puzzle piece that fits with the first one. So here we have a little model of an enzyme molecule, which has a rather nice jigsaw puzzle piece along the top. And here is a molecule with which this is going to interact. This is rather a simple molecule. It's, at the moment, a black and white molecule, and it's going to be broken by breaking a chemical bond into 2 pieces. Now, the first step is that this molecule fits onto the enzyme very exactly in this jigsaw puzzle way, like that, so that it's a very nice, snug fit. And when it's done that, then this piece can fall off separately, because the enzyme has somehow, in a way that we shall see later on, promoted a chemical reaction here, and then this piece can fall off. So having started with one molecule, we end with two parts of this molecule. So shape is the important thing about enzyme molecules. What we want to see is what are their shapes, and how do we find that out? Well, what we need to find that out is a very good microscope, because you'll remember that these molecules are about 100 million times smaller than these models that I keep showing you. So we want a very good microscope. So let's have a quick look at a microscope. Here's a very nice microscope, a Zeiss microscope, in which we are going to look at something. At the moment, we're going to be looking, first of all, at a human hair. But we must wonder, in a little bit of detail, how microscopes work. They have a light down here, which shines up at an object, like a microscope slide, which we put on the stage in this position. Then the object scatters the light in all directions, and the light is captured by the lens of the microscope here, and that focuses it to form an image, which I can look at through these eyepieces. And if I look through there, what I see is a human hair. But if I move the eyepieces out of the way, then this little television camera can have a look at what I was looking at. So if I move that out of the way, I hope it's still there, we should be able to see on the monitor the human hair. And I think that's fairly well in focus. That's a human hair. We shall find out in a moment just about how thick it is. Now, underneath this hair, on the same slide that we're looking at, is a pattern of fine lines ruled close together. So if I change the focus here, and I can just watch the monitor, I hope, as I do that. If I change the focus very slowly, this pattern of fine lines ought to come into view. There we are, pattern of fine lines. It's a slightly dirty slide, so in addition to the lines, there are various patches of dirt on it, but underneath there are fine lines ruled parallel to one another. Now those lines are so close that there are 1,000 of them in every centimetre. So 1,000 lines per centimetre. The hair, if you remember what the hair looked like, covered about 10 of those lines, so it was about 1/100th of a centimetre in diameter, something like that. Now, let's look again in detail at the stages of producing this image. The light comes from down below, strikes the object, is scattered by it. The scattered light is collected by the lens, which focuses it to form an image. Let's look at that in detail, remembering that it's the individual lines on this ruled grating, as we call it, which scatter the light so that each line acts as though it was a source of light itself. Now, light we have to think of in terms of waves. So Bill is going to set up an apparatus in which he can generate water waves. This is called a ripple tank. And dipping into the water here, he has a probe which he's going to make oscillate up and down. So if we run the lights down, now we begin to see on the screen that the source of these waves is about here.
There we are. Got in now.
And when the water is all otherwise settled down, you see that this is acting as the source, and some circular waves are running away from it. That's, let's suppose, one line on the object we were looking at. If we now start another source near to it, the other source is now down here, this produces another set of waves. Now these waves are up and down in the water, and you'll see now that these two sets of waves interfere with one another, so that where there is a peak in the one set and it coincides with a peak in the other set of waves, there you still get a peak. But where there's a peak in one set coinciding with a trough in the other, then they cancel one another out. So you get waves running in some directions, then a dark band and a wave nothing in another direction. Now that's what happens when we shine light at this grating, at these parallel lines, but let's look at that in a little more detail in a diagram. So if we take away the ripple tank and now look at this diagram, this is what we were seeing. Well, to keep it the same way around, I'd better do it like that. Now imagine that light is coming up and falling on one of these lines that we were thinking of, and this line is acting as the source of the light waves, and these are the circular waves coming away from this line. If now we put down another source nearby, say there, which is producing another set of waves, these two sets of waves will be interfering with one another. So that to begin with, you can see that peaks come together along these lines. So if I put down a little bar there and there and there and there, then we have waves running out towards me in the same direction as the original waves. That's, if you like, the wave going straight through. But also notice that over in this direction, this peak is coinciding with this peak, so we get another set of waves coming out in this direction. If I straighten that one up again, if I can. So we have another set of waves coming in that direction, and then over here we get another set coming in this direction. And we get the same thing, which I'm not going to put in, over on the other side of the drawing. Now we can look at that by looking at the first stage of the microscope using this laser. This now is light of a single wavelength, and if Bill puts, you see that that's, if we look again at the beginning of that, Bill, the spot of light from the laser appears here. Then if Bill puts in this pattern of lines in front of it, you see that here's the central spot again, and on either side, 1, 2, and on the other side, 1, 2, there are other spots of light. These are the waves that we saw going off to either side of the central wave. Right? Now, suppose we change the distance between the lines, and we put them rather closer together, like that. Now we still get the waves agreeing in this direction. There's the first peak, and there's the next peak, and there's the next, there's the next. But now for the wave that's coming out over here, it's gone round to this position. There's the first, and there's the next, and there's the next. And the next one after that, the other wave, I can't see any longer. So that this wave is now at a much greater angle to the central wave than we were to begin with. Now, Bill can show us that again with the laser. If we go back to the first pattern, here's the central spot, and here are the spots on either side corresponding to these waves. If he now changes to a grating in which the lines are much closer together, then these spots become much further apart. So the closer the lines are ruled together in this grating, the further apart these scattered waves become. Now, what do we want to do if we want to form an image in our microscope? What we do is put in a lens. There's a lens. And this lens collects these waves and focuses them to form the image. Well, in the one that I've drawn, it could collect this wave, but that's just the wave that was falling on the object. It hasn't got any other information in it. The lens isn't big enough to collect this wave, which is going out in this direction, so it doesn't have any information from that, and it doesn't form an image that tells us anything. And the closer together the lines are, the further round this wave goes, so that when the lines are as close together as the distance between the peaks in the waves, a lens can't possibly focus to form an image. So that sets a limit on how small a thing we can actually see in our microscope. What do we have to do then if we want to look at something which is 100 million times smaller than these very small models we've been looking at? We need to use a radiation with a very short wavelength. X-rays are such a radiation. The wavelength of X-rays is about right for us to be able to see atoms that are joined together. So, all right, we have a microscope, and in this microscope we have to use X-rays. Well, that sounds quite straightforward. So we take a pair of tweezers and we pick up a molecule and we put it on our microscope stage. But there again we have a difficulty because the molecule is really rather too small. How do we get around that? Well, we get around that by using crystals. We can't handle one molecule. Let's have a lot of molecules, and let's have them in a crystal. Here's a model of a crystal in which each blue ball represents a molecule that we want to look at. And these molecules are all regularly arranged in the crystal, all pointing the same way. So we look at this model using our X-rays. And you can see perhaps why I was talking about lines now, because here we have sets of lines in this crystal. So we would expect it to scatter X-rays in rather the way that our lines were scattering light, except that we've got lines going this way as well. So here we have a rather more complicated grating. It's got lines going up and down, and it's got lines horizontally as well. And we'd call that a cross grating. How would we expect that to scatter light? Well, most of you, I think, are carrying cross gratings in your pockets. If you like to take out a handkerchief and look at this light that Bill is putting up through your handkerchief, then I think you might see, especially if you've got rather a good handkerchief, you might see the pattern into which the light is scattered by your handkerchief. And if we put a handkerchief over the television camera, then on the monitor you'll see that as well. And if we turn it round, you'll see that the pattern turns round. The pattern is somehow coming from the object. So that's how we would expect a cross grating to scatter light. And since we saw that a crystal looks, at least in one direction, it looked as though it was like a cross grating, then if we put a crystal into an X-ray beam, we would expect to see a somewhat similar pattern. Well, Bill has a cross grating, and he can put that into the laser beam, and then you'll see the pattern. This is now using light of a single wavelength, and there is this rather pretty cross pattern of spots produced by the scattering of light from this cross grating. Now suppose I put a crystal into an X-ray beam, and I put a film behind that to record the pattern of spots from the crystal, and here is such a film. Then we'll see what sort of pattern is produced by a crystal and X-rays. And I think you can see that the pattern that we produce from the crystal and the pattern produced by the light from your cross grating, these two patterns are very much the same. So we are making progress. What we do next is to put a lens into the system, and if Bill now puts a lens in front of his pattern, and we run the lights down again, you'll see that the pattern of scattered light is now replaced by an image of his cross grating, the handkerchief pattern. If you take that away again, there's the scattered light pattern, and if you put the lens in, that focuses that pattern to form the image of the cross grating or handkerchief. So all we have to do is, thank you very much, Bill, is to put a lens into our X-ray microscope and produce the image of the crystal. But there's a problem. We don't have a lens that will focus the X-rays to form an image like that. So what do we do about it? Well, what we have to do about it, instead of putting the lens in, we have to measure all of the information, all of the spots in photographs like this. And you have a different photograph like this for every way of looking at the crystal. You measure the positions of the spots and how black they are. Now that was first of all done, in effect, by W.L. Bragg, who was director of the institution here immediately before Sir George Porter. And he used quite a lot of the time an apparatus which was designed by his father, W.H. Bragg, who was a director of the institution earlier on. And they used an apparatus like this, where this is a rather old X-ray tube. Just imagine the safety hazards of using an X-ray tube like that, with X-rays coming off in all directions, striking the people, whereas all you want them to do is the X-rays to pass down this little tube here, so that the X-rays strike the crystal at the middle of the apparatus. Now with this apparatus, what they did in effect was to measure one of those black spots at a time. So they would set this detector, this is an ionization chamber detector, at the right position to measure one of the black spots and measure its strength, and then move it all so that they can measure another black spot, in effect, and so on, until they'd measured completely the way in which this crystal scatters the X-rays, and then from that you need to build up the image of the structure. How do we build up the image of the structure from that pattern? Well, I can indicate what's involved in that if we look at a slide. On this slide, we have just a diagram which represents this pattern of black spots you've been looking at. Here's the center. This open circle is the center where the X-rays strike the film. This spot is the nearest to the center in that direction. This spot tells us that in the crystal, and we have to note here that it's the electrons in the crystal which are scattering the X-rays. In the crystal, these electrons form bands running in this way, so that in that distance there is one wavelength. There is a positive region here, a negative region between them, and a positive region again. There's a wave there with one wavelength in that distance. This spot tells us the wavelength and the amplitude of that wave. The next spot tells us that there's a wave with half the wavelength running in that direction. The third spot tells us there's a wave with a third of the wavelength. So this is a sort of harmonic analysis of the way in which the electrons are arranged in the crystal. This spot tells us there's a wave running that way. And these spots tell us there are waves with shorter and shorter wavelengths running that way also. This spot on the diagonal tells us there's a wave running in that direction, and so on. What we have to do to build up the image is to add all these waves together, and that will give us the image. But there is a difficulty. If you add them all together the way they're drawn here, we shall have lots of electrons at the corner of each pattern, and they will all add together, and at the corner there will be an enormous concentration of electrons. That wouldn't be right. The problem is, do we put the positive corner of this one in the same place as the positive corner of that one, or at some other position? Perhaps the positive corner there has to go at the middle of this white region on the next wave, and so on. We don't quite know how to put these waves down in register. That's the difficulty. If we knew how to do that, we could build up an image, and we would do it in this way. Let me illustrate it with a simple structure. Here we have one wave running through such a structure. Here are the peaks in the waves. The electrons, as far as this wave is concerned, are mostly in these regions. If we put down another wave, this wave is running this way. The electrons are mostly in those regions. Then another wave, and another wave, and another wave, this one running this way, you see. And slowly, there, we are building up an image of the structure. And what we can see is that there seem to be concentrations of electrons in this regular hexagon pattern. That's actually the build-up of the structure of benzene, which is a six-membered ring. If I put that on top here, that will be telling us the structure of benzene. So to build up images, what we have to do is to put all of this information somehow into a computer, and that tells us the structure. The computer, given all this information, and more information to solve the problem that I mentioned, and it was Max Perutz who first showed us how to do that with a protein. This computer produces tables and tables of numbers like this. And the numbers are telling us how many electrons there are at each particular position in the crystal. But we can't look at that and make much sense of it. We have to do something to make sense of it. What we now get the computer to do is to draw contour lines, so that it now is drawing lines through positions where there are the same number of electrons. So this little mountain range you can see here means that in that region there is a concentration of electrons or atoms. But the image we need to make is a three-dimensional image. How do we make it three-dimensional? Well, the computer calculates such sections throughout the structure, and we put these sections onto transparent sheets, There's the one we've just been looking at. And we stack it up together so that we form slowly a three-dimensional image. There you see we are building up an image. And if we look at this closely, you see that we are seeing lines of protein chain running in these directions, and that is very much the structure of silk that we looked at last time, where the protein chains are drawn out in straight lines and they are joining themselves together by these electrical connections. That's the sort of image we can build up of a protein molecule with that silk-like structure in it. And here we have another image of a part of a molecule which has a different kind of feature in it. Here, this feature. And if we look at that rather strong feature, and if I rock this, you can see that more closely, then this is, when interpreted in detail, the same structure as the structure of hair that we were looking at again last time, this spring-like spiral structure. Thank you. So if we look at all these maps, we can derive from them the three-dimensional structure of protein molecules. This was first done for an enzyme structure here in the Royal Institution about 15 years ago, and I was lucky enough to play a part in that with a group of my friends, and my friends are shown on this slide. Here you see them, from left to right: Gareth Mayer, Colin Blake, Louise Johnson, Tony North, and Raghu Sharma, and together we produced the structure of the first enzyme ever to be looked at in this detail. Now, what enzyme was that? It's an enzyme that involves a rather famous person. It was an enzyme first discovered by Alexander Fleming. And Fleming is shown on this slide, taken, that's a photograph taken at about the time when he did the work, which began with a rather strange experiment. Fleming was a person who studied bacteria, germs, if you like, and he was anxious to find a way of killing germs without killing the people that the germs were infecting. So he spent a lot of time growing germs on culture plates. That's to say, little plates like this, which would have a nutrient broth in them here on which the germs would grow. Now, Fleming was, to say the least, an interesting man, and he did interesting experiments. And one day in about 1920, he had a cold. And he had a lot of these bacterial cultures lying around. So he picked one of them up and let his nose drip onto it. And then he put it down again and went home for the weekend, perhaps to go to bed. And when he came back, he looked at it again. And imagine his surprise, because when he looked at it again, what should he see but a pattern like this? In this culture, and this is such a culture, the cloudy white color is the bacteria growing, and the blank patches, which have a reddish brown color of the nutrient underneath, these are the spots where the nose drops had fallen, and there the bacteria seem to have been killed. He got very excited about that. Here was substance produced in his own body which killed bacteria. So he quickly looked at other things, at blood, and in particular he looked at tears, because he thought the eye was a good place for bacteria to grow. And he found that human tears had this effect also of killing some bacteria. And this led to some excitement. There was a view that perhaps this substance could be produced commercially, and the design for a factory appeared in the London evening papers, as you see in the next slide. The idea was that by getting tears from volunteer children by some appropriate method, one could collect the tears and then use them as an antiseptic in this way. Well, that's the substance that we study. It turned out that this is an enzyme, and he called it lysozyme, meaning an enzyme that dissolves bacteria. It turns out that it can be crystallized. And here is a model of the sort of crystal that you produce. You see, it's only a model, but I must assure you the actual crystals are just as beautiful. They're much smaller. A rather beautiful pattern, and you see, looked at that way, it has this squared arrangement that you saw in the photographs, and indeed that X-ray photograph was a photograph of lysozyme crystals. Now, the actual crystal is a good deal smaller, and here I have one in a specimen tube, because the crystals contain a lot of water, and you have to stop them getting dry by putting them inside a glass tube together with some water. And if we look rather closely at this specimen, it's in a glass tube between two pieces of plasticine, and in the middle where the arrow points, you might just pick out a little spot of light which is shining from the very tiny crystal that's in there. That's how we have to mount these crystals on an apparatus something like the one we were looking at in order to make all these literally hundreds of thousands of measurements. But having made all those measurements, then at last we have the ability to build the structure of lysozyme, and here is a model of it. Here we have a model of the lysozyme molecule. Now, unlike the other molecules that we've been looking at, it isn't long and stretched out the way silk was, and it's not long and stretched out the way in which hair and tendon were. It's a compact folded up molecule with this rather interesting shape, a little bit like a rugby football, except that on this side there's a deep groove just there where I can put my hand. Now, you have to note that the protein chain in this molecule is the same as the protein chain which is hanging up there at the back. So that long chain is folded up to form this compact structure. Why does that long chain take up this particular compact structure? Well, that's because of the order in which the components are arranged in that long chain. That's because of the arrangement of these components, and if we abbreviate them in the usual way by letters, that's far enough, I think, almost. There we are. In this particular chain, there are 129 of these components. And you see the letters there seem to be in a quite random arrangement. There are 20 different letters. If we could look closely at it, there is one of each of those at least, arranged in a rather random way. That particular order of the protein components is what decides that this particular chain is going to take up this particular three-dimensional shape. Now, we'd expect this shape to have something to do with the effect on bacteria. And now we must look at the nature of the bacteria. Here's a diagram that shows schematically the chemical structure of the wall around the bacteria. And you see at the top here we have a row of green hexagons. These are molecules we haven't met yet. These are sugar molecules arranged in a polymer chain, and there are two sorts which we've labeled here N, M, N, M. every other along the chain. The difference is that the M ones have a little protein chain attached to them, and these protein chains, they reach down eventually and connect to another string of sugar molecules down there. Now, lysozyme is able to break one chemical bond in this structure just there, this particular chemical bond. Next to an M and before the oxygen, lysozyme can puncture the wall of a bacteria. A bacteria is a single cell organism, can exist as a single cell which has this wall around it. This wall around it is made in this way, and lysozyme can puncture it by breaking that one chemical bond. How does it do that? Well, happily, we are able to build into our model of lysozyme a part of this bacterial wall structure. And in this model, you can see that we've added it in this rather pretty structure of hexagons. If we look at this in a little detail, up at the top here is one hexagon. This is one of these sugar rings which were colored green, and it's one that we called N. The next one along, a little bit lower down, I've attached to that this rather large black ball here. There's a black ball just there, stuck onto the second one. This is an M-type sugar ring, and this black ball represents the place where the protein chain was attached in the diagram we were looking at. So if the protein chain is out here, this must certainly be on the surface. The next one, which you can just see in there, is a hexagon again, that's N. The next one down has a black ball again on it, that's M. The next one down, here's a hexagon, that's N. The next has a black ball, that's M again. So this set of sugar rings joined up in a polymer in this way fits exactly into this groove in the enzyme structure. If I turn the model, you might be able to get a better three-dimensional effect than you can just by looking at it in the distance. How do the two fit together? Well, the sugar molecule and the enzyme make a lot of these electrical connections, plus-minus interactions between parts of the sugar molecule and parts of the enzyme. If we look, for example, down at the bottom here, then between the sugar at this point and the enzyme here, here is a nice plus-minus interaction. Up here is another plus-minus interaction. And throughout this structure, there are a lot of interactions of that sort. So there are these plus-minus interactions. And then there's the general shape. The two really do fit together like two jigsaw puzzle pieces. A jigsaw puzzle piece made of the sugar part of the bacterial cell wall, and the gap in the enzyme structure is the part that that fits into. Two jigsaw puzzle pieces fitting together. What about the bond that is going to be broken? Well, I don't know how easy it is to see here. It is, in fact, a bond between the fourth and fifth of these sugars. It is a bond just there. The red ball is an oxygen, and the orangey ball just above it is a carbon. It's that bond that my finger is just resting on. Somehow, the enzyme brings about the breaking of that bond. Now, next time, we shall try to look at how the enzyme actually succeeds in doing that. But for the moment, we won't do that. We'll have a look at the bacteria. But before we look at the bacteria, there's just one other little thing I'd like to do in preparation for a later lecture. It turns out, unhappily, that the bacteria which lysozyme kills are not very harmful. And that may be because we produce lysozyme and are therefore able to kill them. Because the fact is that we live with bacteria all the time. And what I would like to begin today is an experiment to see something about the bacteria we live with. So we're going to pass around some, or hand to you, some culture plates for growing bacteria. And I'd like a few of you to put thumbprints on them. I'm sure your hands are very clean, but never mind. And I'd like some more of you perhaps to kiss them, if you wouldn't mind. And then a little later on, we shall see whether any bacteria have grown on these cultures. And we'll use that in the next lectures. You'll notice that the plates have to be covered up very quickly so that the bacteria in the air don't get onto them and spoil this culture that we may be growing. Have we had a few kisses as well as thumbprints? Good. Thank you very much indeed. Now, what does lysozyme do? It punctures this bond in the bacterial cell wall. Let's have a model of a bacterium. Here we have a model of a bacterium. And on it, we've drawn this chemical formula. What the lysozyme is going to do is to burst this bacterium by breaking just one chemical bond in it. In fact, no doubt, in practice, a lot of lysozyme molecules would approach one bacterium at the same time and make a lot of holes in it. But one will do, perhaps. The chemical bond that's going to be broken is between this sugar ring, let's say, which is the one carrying the protein chain, that's an N-type sugar ring, and the oxygen atom attached to it. So lysozyme is going to puncture just this chemical bond here. Now, some of you have got perhaps some enzyme in your pockets. I don't know. What I'd like would be one or two people who have got some enzymes to come up and see if they can puncture these bacterial cells. Here we are. Here are some bacteria which are now floating into the lecture theatre. Let's see if you could manage. That's right, but you have to puncture it in the right place. No, no, no. You have to look for the right place to do it. Right? That's right, good. No, no. Try again. No. How about there? Right. Right. There we are. And the bacteria get killed in that way. Next time, we'll find out exactly how the lysozyme does it.